Assalamu alaikum. Today we are going to start the fourth chapter of biology, that is cell biology. It contains information that what is the cell, how it works, what are its organelles, and all the information about this cell we will study in this chapter. The first thing is about the microscope. There are actually two types of microscopes. The first one is the light microscope and the other is the electron microscope. How they work, that the light microscope actually contains the two glass lenses, and it works by passing the visible light through the specimen. Like there is the object present and through which the visible light can pass, and it helps to see the object. Its magnification power is about 1500 times and its resolving power is 0.2 micrometer. Along with this, the lens produces an enlarged image of the specimen, and it has the other lens, the second lens, magnifies the image and it projects into the viewer's eye or onto the photographic film, where we can see, and a photograph taken through a microscope is called as the micrograph. The other thing is about the electron microscope. Here it is written that object and lens are placed in a vacuum chamber and beam of electrons are passed to the object. So in this way, electron passes or reflect from the object and it makes the image of that. So electromagnetic lenses enlarge and focus the image onto a screen or a photographic film. So electron microscope has actually the much higher resolving power than the light microscope, and the most modern electron microscope can even distinguish the object as small as 0.2 nanometer. That's why we say that resolving power is 0.2 nanometer, while its magnification power is 250,000 times more than that of the light microscope. Here is a figure, light microscope, and this is the electron microscope. This is the simple one, while the light microscope is a larger one. It can even see the internal structures of the cell. Electron microscope is also divided into two more types, that is transmission electron microscope and scanning electron microscope. This transmission electron microscope it uses transmitted electrons. Transmitted electrons means electrons which are passing through the sample to create an image, and it is also used to study the internal structures, while scanning electron microscope creates an image by detecting the reflected electrons, and it is used to study the structure of cell surfaces. Now next thing is about the history of the formulation of cell theory. There are different scientists to give different theories about the cells, according to which the first one is about the Z. Jensen. What he said, he invented the microscope. Then the second one is about Robert Hooke. Robert Hooke in 1665 discovered cell. In 1674, A. V. Leeuwenhoek, he observed the microorganism to the microscope. Then J. B. Lamarck in 1809 proposed the importance of the tissues. In 1831, Robert Brown discovered the nucleus. Then the plant tissues were studied by Matthias Schilden in 1838. In 1839, animal tissues were studied by the Theodore Schwann. Then 1855, omnis cellulose cella was proposed by R. Virchow, and the Virchow's idea was proved by Louis Pasteur in 1862. This is all about the history of the formulation of cell theory. Now, what is the cell theory? Cell theory actually tells us it is about different principles, in which the three main are that all organisms are composed of one or more cells. Cells are considered as the smallest living things and the basic unit of organization of all living organisms. Then they also say that cells arise only by divisions in previously existing cells. Like if some cells are present, then these cells can divide and produce the new cells. Then we come towards the cell structures, its functions, and its organelles. The first part is about the part of the cell is cell wall. Cell wall is the non-living strong component of the cell. It is present outside plasma membrane, means it is present on the outside of the outside layer, that is means the cell membrane is considered, or plasma membrane is considered as the outermost layer. But in case of the plant cells, it is the cell wall is present outside the plasma membrane. It helps to maintain the shape, strength, production, and support to the living matter of the cell. It is also primary. There are two walls, that is the primary wall and the secondary wall. The primary wall, the outer layer of the plant cell is known as the primary wall, and cellulose is the most common chemical in it. Some plant cells, for example xylem and cells, have the secondary walls on the inner side of the primary wall. It is much thicker and contains lignin and some other chemical. These are pores in the cells of the adjacent cell, and these pores are called as the plasmodesmata. As we said that it is made up of the cellulose, but in many fungi and protists, cell walls, although they do not contain cellulose, so their cell walls are made up of variety of chemicals. For example, chitin is present in the cell wall of fungi, and in prokaryotes it is made up of peptidoglycan. Then we come towards the next part, that is cell membrane. Cell membrane is actually the covering of cytoplasm. It is elastic, semi-permeable barrier. Semi-permeable barrier means it allows some of the substances to pass through it, which are good for the cell, and it does not allow few substance molecules to cross the membrane and move inside the cell. In this way, cell membrane maintains the internal composition of the cells. It is composed of lipids and proteins. Here is the fluid mosaic model of the cell membrane. According to the fluid mosaic model, actually what that chemical analysis reveals that cell membrane is mainly composed of proteins and lipids with small quantities of carbohydrates. So electron microscopic examination of cell membranes have led to the development of this model, which is called as the fluid mosaic model of cell membrane. According to this model, there is a lipid bilayer, means there are two layers of lipid. As you can see in the figure, these two yellow layers are actually the layers of lipid, in which the protein molecules are embedded. The purple ones are actually the protein molecules that are embedded into the lipid bilayer. Small amount of carbohydrates are also found in the cell membrane. Here you can see these are the small amount of carbohydrates that are also present in the cell membrane, and they are joined with proteins or lipids of membrane. You can see that these are joined with the lipids of the membrane. So this is actually the fluid mosaic model of cell membrane. Then cytoplasm. Semi-viscous and semi-transparent substance. It actually contains all the organelles of the cell. So it contains water, in which organic molecules and in organic salts are completely or partially dissolved. What are the organic molecules? The proteins, carbohydrates and lipids. It is also the space for the proper functioning of the organelles and for the various biochemical functions. For example, the glycolysis, that is the breakdown of glucose during cellular respiration, it also occurs in the cytoplasm. The next thing is cytoskeleton. Cytoskeleton is what? That it is the network of microfilaments and microtubules. Now these are the two types, microfilaments and microtubules. Microtubules are made up of tubulin proteins. You can see these are the microtubules, and these are tube-like structure. These are made up of tubulin protein, and it is used by cells to hold their shape. While microfilaments are made up of actin proteins, and it helps to change the shape of the cell. You can see these are the microfilaments. The next starts for the further organelles of the cell. The first one is the nucleus. Nucleus is present in the center in the plant cell, animal cell, while in case of the plant cells it is present near to the cell wall. The reason is that in plant cells a large vacuole is present, due to which the nucleus moves towards the membrane. Then a nuclear envelope is present. What is it? That nucleus is bounded by a double membrane, that is called as nuclear envelope. It contains many small pores that enable it to act as a semi-permeable membrane, and inside the nuclear envelope a granular fluid is present. This granular fluid is called as nucleoplasm. Then a dark spot is present. Here you can see this is the nucleolus. It is a dark spot, and it is the site where ribosomal RNA are formed and assembled as the ribosomes. Then you can see these are the chromatins are present. These chromatins combine and form the chromosomes. And these chromosomes are actually composed of DNA, that is deoxyribonucleic acid, and proteins. The prokaryotic cells do not contain the prominent nucleus. Their chromosome is made of DNA only and is submerged in the cytoplasm. So the next organelle is ribosome. Ribosomes are actually the tiny granular structures that are freely floating in the cytoplasm and are bounded with the endoplasmic reticulum. Each ribosome is made up of almost equal amount of proteins and ribosomal RNA. These are made up of the proteins and ribosomal RNA, and they are not bounded by any of the membrane. These are considered as the site for the protein synthesis. And protein synthesis is extremely important to the cells. So large numbers of ribosomes are found throughout the cells. When the ribosome is not working, it disassembles into the smaller units. Otherwise, it is present in the larger unit and it helps in the formation of proteins. Next organelle is mitochondria. Mitochondria is actually called as the powerhouse of the cell. So double membrane bounded structure it has, because you can see this is the outer membrane, this is smooth, while the inner membrane makes enfoldings. These enfoldings are called as cristae. Cristae. These are the sites of aerobic respiration and the major energy production centers. Then this serves to increase the surface area of the inner membrane. These enfoldings are increasing the surface area for the inner membrane. And mitochondria have their own DNA and ribosomes. The ribosomes of the mitochondria are more similar to bacterial ribosome than to the eukaryotic ribosomes. Next one is plastids. Plastids are also the membrane-bounded organelles that are occur in the cells of plants and photosynthetic protists. Means these are not present in the animals. They have the direct link with the photosynthesis, and these are only present in the plants. It is of three types: chloroplast, chromoplast, and leucoplast. There are the sac-like structures present here. You can see that these are the sacs. These are called as thylakoid. And then the stack of thylakoids is called as granum. This one is called as the thylakoid, while this whole stack is called as the granum. So three types: chloroplast. Chloroplast is actually, it gives the green color to the plant. It is involved mainly in the photosynthesis of the plant. The other one is the chromoplast. They contain chlorophyll and associated pigments. These pigments are present in the thylakoids of grana, and they contain the pigments which are also associated with the bright colors and represent the cells of the flower, petals, and fruits. While the third one is leucoplast. Leucoplast is the third type, and it is colorless, stores starch, proteins, and lipids, and are present in the cells of those parts where food is stored. Next organelle is endoplasmic reticulum. Endoplasmic reticulum is the network of interconnected channels. It has two forms, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is so named because of its rough appearance. As you can see in this figure that these ribosomes are present on it. That is why it is giving the rough appearance, and it is called as the rough endoplasmic reticulum. Then it serves as a function in the protein synthesis, while the other one is the smooth endoplasmic reticulum. Smooth endoplasmic reticulum, what it did, it does not contain the ribosomes, and that is why it is called as the smooth endoplasmic reticulum, and it is involved in the lipid metabolism and the transport of materials from one part of cell to the other. It also detoxifies the harmful chemicals that have entered the cell. The next thing is about the Golgi apparatus. The Camillo Golgi, the first time we discovered the Golgi apparatus, these are the flattening sacs called cisternae in the cell. These sets may, in this set, many cisternae are stacked over each other. You can see these are all stacked over each other. And the complete set of cisternae is called as the Golgi apparatus. These are found in both plant and animal cells, and it modifies molecules coming from the rough endoplasmic reticulum. And what it did is that then packs them into the small membrane bound sacs, which are called as the Golgi vessels. And these sacs can be transported to various locations in cells or to its exterior in the form of secretions. Next one is lysosome. Lysosome was first time discovered by Christian and Jean D. Dove in the mid-20th century. They are single membrane bound organelles. Lysosomes contain strong digestive enzymes, and it works for the breakdown of food. So lysozyme actually how it works, that it fuses the vacuole that contains the targeted material, and its enzymes break down the material, and in this way these lysosomes can be formed and work. Okay, the next one is centrioles. Centrioles are the hollow and cylindrical. It is the 9 triplets of molecules. You can see this is the triplet. These are the 9 triplets of the microtubules. And then the 2 centrioles are collectively called as the centrosome, and their function is actually to help in the formation of spindle fibers during cell division. In some cells, the centrioles are involved in the formation of cilia and flagella. Next thing is vacuole. The vacuole is actually the fluid-filled single membrane structure. Cells have many small vacuoles. In plant cells, a single large vacuole is present. Cells in this state becomes the turgid when you can see here the large vacuole. If the large vacuole is present in the center, nucleus is present on the side, it makes the cell turgid. And many cells takes part in materials from outside in the form of food vacuole and then digest the material with the help of lysosomes. Some unicellular organisms use contractile vacuole for the elimination of waste from their bodies. And this is all about the organelles of plant. The next thing is what is the difference between the prokaryotic and eukaryotic cells. If we talk about the nucleus, in case of the eukaryotic cells, their prominent nucleus is present, while prokaryotic cells do not have prominent nucleus. Their chromosomes consist of DNA only, and it floats in the cytoplasm near center. This region is called as nucleoid. The other organelles, in case of the eukaryotic cells, the membrane-bonded organelles like mitochondria, Golgi apparatus, endoplasmic reticulum are present, while such membrane-bonded organelles are not present in the prokaryotic cells. So the ribosomes of eukaryotic cells are larger in size as compared to the ribosomes of the prokaryotic cells. In size, eukaryotic cells are on average 10 times larger than the prokaryotic cells. Then the cell wall of eukaryotic cell is made up of cellulose or chitin. All prokaryotic cells have cell wall, which is made up of peptidoglycan, a large polymer of amino acid and sugars. Then what is the relationship between the cell function and cell structure? For example, if we say about the size and shape, the red blood cells are round to accommodate the globular hemoglobin. Nerve cells are long for the transmission of nerve impulses. Xylem cells are tube-like and have thick walls for the conduction of water and support. Then the surface area, the root hair cells have large surface area for the maximum absorption of water and salts. Presence or absence of organelles, the cells involved in making secretions have more complex endoplasmic reticulum and Golgi apparatus, and cells involved in photosynthesis have the chloroplast. In this way, the presence of organelles have the major role in the functioning of the cell. This is all about the organelles and their functioning, and we will study about the tissues in the next lecture.